Hi everyone, welcome to my journal review. My name is Hadi Fal, and I'll be talking about microbial signatures associated with glycemic control in type 1 diabetes. Today, this we'll be talking about this review, which is a study by Schill et al., and it explores the relationship between the gut microbiome and glycemic control in adults with long-standing type 1 diabetes. This is a slide that's prepared for the acronyms that we will be coming across in the presentation. So in regards to the bluff and the study rationale of the article, why did the scientists, um, you know, get into this research? Why should we care about this field in general? And did they achieve anything at all? I would say this study was very pertinent because it talks about how adults with type 1 diabetes have distinct gut microbiomes compared to like a healthy cohort, and how gut microbiomes can vary in terms of glucose variability and offers potential therapeutic targets in the future. The study also fills a critical gap by focusing on adults with long-standing type 1 diabetes and aiming to understand, how their gut microbiome composition, how it functions in relation to the glycemic control, the blood sugar level, etc. And what is the main association between the microbiota composition and the pathogenesis of type 1 diabetes? Also, glycemic control in adults is less studied than those of pediatrics. This is the adults with type 1 diabetes versus the pediatrics with type 1 diabetes. I would say the present scientific question is, whether gut microbiome profile in type 1 diabetes patients can distinctly be characterized, and can it be correlated to host glycemic control? I will say this question is pertinent because type 1 diabetes is a lifelong condition with increasing global prevalence and holds significant health risk. The study will help understand gut microbiome associations and could reveal, non-invasive markers or therapeutic targets to enhance glycemic management and prevent complications with people with type 1 diabetes. In general, I believe the study was successful, but further studies will be needed to identify particularly how this bacteria in the gut microbiome play, what role do they play in potential therapeutic strategies. Just a quick background of the global prevalence of diabetes in the world. As the chart shows and summarizes, diabetes is prevalent globally, increasingly worldwide, and the highest concentration of new cases remain in Europe and America, and Asia is rapidly rising too as well, which if reflects like increase in diagnosis rates, probably better reporting, and maybe lifestyle shifts. Latin America and the Caribbean, in combination with Africa and Middle East, collectively represent 30% of new cases, in fact, emphasizing a new burden of global disease. Here also represents the estimated number of people with type 1 diabetes by country and age distribution in the United States. I would say from this observation, we can say the growing incidence of diabetes in middle-income countries especially highlights the need for improved diagnostic infrastructure and public health strategies. So what is the urgency of this study? As I already talked about, diabetes is a high, it's becoming like a global disease burden, and it's getting like highly prevalent in most regions in the world. And most of the time, patients result in lifelong insulin dependence. And, it's usually diagnosed in childhood, adolescence, or early childhood. That is, if the resources are available in those regions. Ongoing research is going on in immunotherapy, stem cell transplantation, and particularly modulating the gut microbiome as a potential intervention, which is in regards to our talk today. Here, the image talks about how different factors can contribute to the microbiome, and then eventually lead to the gut microbiome, eventually leading to type 1 diabetes of a patient. This includes genetics, the environment, microbial infection or viral infection, diet, antibiotics, some sort of inflammation, and the dysbiosis of the microbiome, which is a shift in the abundance of the gut microbiome, and correlates or result in altered functionality, such as the reduction in production of like short chain fatty acids, um, and eventually disrupting the gut microbiome. So what is the article objective? The main objective of the study, like I mentioned previously, is to study the relationship between the gut microbiome of type 1 diabetes patients in adults with the glucose variability, glycemic control, and how, the results of this study can offer potential therapeutic targets in the future. And it mainly focuses on adults with long-standing type 1 diabetes versus pediatric diabetes, and how, it can function and relate to glycemic control. So talking about the study design, I'll be talking about this and also talk about it later during the methodology, as it's part of the methods of the study. So the clinical cohort originally was designed for to study the PPGRS in type 1 diabetes, which is the postprandial glycemic responses of individuals in type 1 diabetes. And this is a measurement of the blood glucose levels after eating, revealing how your body processes carbohydrates and how it uses insulin compared to like fasting glucose alone. Anthropometric measurements of candidates were also done, which is the scientific study of the human body measurements for a better cohort match. Blood tests were also drawn, and health and lifestyle questionnaires too were completed by participants for the 2-week study duration. I will point out this study was done in Israel in a medical center, and I will say they did a lot of work to get a good cohort match between healthy patients and type 1 diabetes patients. I feel like it's a great model, research experience, which can be used in different countries as well, not just Israel. Also, they provided participants with a nutritional smartphone app to, log in their daily lifestyles, like in real time, like food intake, sleep times, physical activities, and medications, except insulin, which was recorded in a different device. And participants were asked to, follow their normal routine and dietary habits, and also give out a sample, that is a stool sample. Here is a quick overview of the study design. It's more like a timeline snapshot sort of the study design. They started with 142, patients enrolled in the study and ended up with 74. And then they had like 35,000 controls, healthy controls. I feel like that's a lot of people. And, because they had to do like really good cohort matching in terms of their BMI, in terms of their age and sex, and, um, before they ended up with this number here. So it's a 1-to-4 matching. So the microbiomes of adults, patients with diabetes, they had to have like 15 years, at least 15 years of diabetes post-diagnosis. And um, the participants were 74 adults with type 1 diabetes and, 296 healthy adults. And for the methods, they used the shotgun metagenic sequencing, continuous glucose monitoring, and blood biomarkers. And comparisons were done between the healthy cohorts and the patients using the LDA, that's the linear discriminant analysis, as well as the FDR statistic approach to control multiple testing and also get a corrected p-value. So in terms of the methods they use, this is a slide that just talks about an overview of the methods they use. Cohort matching was done, stool sample collection, genomic DNA extraction, and Illumina sequencing. So here is a table that shows the cohort summary table, the cohort summary after doing a cohort match. So it talks about the inclusion and exclusions for the um, participants that were involved in the study. So the average ages were between 18 to 70 years old, and you had to have like more than a year, at least more than a year, ideally 15 years following type 1 diabetes diagnosis. The use of continuous glucose markers and continuous subcutaneous insulin infusion devices. And they had to match like, they had to be compatible in terms of their sex, weight, BMI, and, in terms of the patients in the health cohort, the healthy control groups. Exclusion criteria. So they shouldn't have anything that's related to like inflammation or pregnancy or antibiotic use 3 months prior to the study. And the participants with a diagnosis of celiac disease were disqualified because apparently celiac disease is correlated with a change in the gut microbiome composition. They also had a log of the total energy intake and macronutrient intake in terms of carbs, fat, protein, the common medications used, and etc. BMI, HbA1c, that's related to the blood glucose, and the provided stool samples. So in terms of the sample collection, the participants, they were trained to get their stool, like the stool collection instructions had to be followed clearly to avoid any contaminations. Two methods were used, the swab and the OmniGene GOT kit, but only the OmniGene GOT kit samples were analyzed to ensure consistency and DNA stability. And samples were stored in home freezer that's minus 18 before transported to the lab in a minus 80 degrees Celsius. So first we'll talk about the DNA extraction and sequencing process. I will briefly talk about this. I also included a snapshot of the process in general, which was adapted from a different study. They used the metagenomic DNA QIAGEN kit, MegaTrack Power Soil DNA extraction kit. to do the extraction and ended up sequencing the product from the DNA extraction using the NovaSeq Illumina sequencing and estimated the relative abundance using coverage of unique, genomic regions. Here's a snapshot of the process, which is similar to what they did, extracting the stool, vortexing, and ended up removing impurities. So the Illumina sequencing, we talked about this in our class, but I just wanted to include a slide that gives us an overview of what the Illumina sequencing is like in terms of the pros and the cons and why the study used this. So they used the NovaSeq sequencing technology because it's very good to use in like large cohort studies like this one. And it's a state-of-the-art high throughput sequencing platform, and it delivers low cost per base. It generates high quality short reads with a deep coverage, which is really good. And some of the cons, of course, it produces shorter read lengths compared to like the PacBio, which has longer read technologies. And there is also a higher risk of assembly errors. And here is just a summary of what we talked about in terms of the sample collection, DNA workflow, and they ended up with the microbial profiling. And some of the taxa that were monitored after the entire sequencing process were these 4 taxa, and also fecal bacterium prosnitzi. And all these taxonomic assignments were used using NCBI classification standards. Also, in terms of like the statistics they use the alpha diversity measurement. They use the Mann-Whitney U-test statistical comparison and the functional profiling, which relates to this here, functional microbial profiling. Mapping the reads to species level genome beans. Those are the GBS. Talking about the results now, so they ended up like showing like mainly, going back to the question they initially asked, what is the correlationships between like the different microbial stains that we observed, the functional pathways and the clinical phenotypes? For this image here, it shows the different types of functional pathways, metabolic pathways in the bacterial taxa and how they all correlated. This is a heat map, and in this part of the study, the researchers explored how microbial species and their metabolic pathways were associated with clinical health markers, particularly the lipids and the glycemic control. They found several interesting patterns. For example, for this species here, the Prevotelaceae family here, they negatively correlated with the HB1AC, which is the glucose level, blood glucose level, meaning the higher levels of this bacteria were associated with better blood sugar control. On the other hand, the Enterobacter species here, it shows a positive correlation. So the pink means positive and the blue means negative here, with average glucose levels, suggesting a potential link to proper, to actual like lower glycemic outcomes here. So as we can see, you have a positive output here and a lower output here in terms of the Enterobacter species. Also, another key finding was the Clostridiales species and the Firmicutes groups. They were linked to lower triglyceride levels. Here. And when it came to microbial functions, several metabolic pathways, particularly those involving like amino acid and other biosynthesis were associated with like higher average glucose levels, like here. So it's a heat map with a lot going on, but basically like what we can confirm and what the scientists confirmed from this result is, a healthier gut bacteria are linked to like better glycemic controls, like healthier bacteria with the Faecalibacterium prausnitzii has a very high glycemic control index. So it's linked to that, and pathogenic or inflammatory taxa like the Enterobacter had like certain metabolic pathways. They correlate with a lower lipid and glucose profile. And these patterns highlight a possible functional and taxonomic microbial signature associated with like glycemic regulation in type 1 diabetes. Another interesting results was this one that I adopted from the paper as well. And it's kind of like a microbiome composition in adults. It's more like a snapshot of everything that's going on. So it's divided into like 4 panels. Panel A here talks about the LDA, which is the linear discriminant analysis of the taxa. The green bars represent the taxa enriched in healthy controls, while the red bars represent the taxa enriched in the type 1 diabetes individuals. And the longer the bar, the greater the effect size of the LDA score here. And it could be derived that the species like here, for example, Prevotella copri, Eubacterium cerium, and this one, Romney Clostridium, have a more enriched in the type 1 diabetes patients, while for the healthy controls, we have like the Faecalibacterium prausnitzii, Alphips petridennis, and Rhinococcus have like a higher enriched profile. And it shows the distinct compositional differences in gut bacteria between the 2 groups ranked by the effect size, genus, strain, and family. And for B, it talks about the ROC curve for the predictive model. And the mean ROC has this high number of 0.089, which indicates that the gut microbiome can reliably distinguish type 1 diabetes individuals from healthy controls. So there's a clear difference between those 2 different cohorts in regards to their microbiome composition. And we have the Shannon diversity panel as well, which doesn't actually show any statistical difference between the, type 1 diabetes and the healthy control. There's like a difference, but it's not like very significant. And it concludes that the overall bacterial diversity is not a major distinguishing feature between these groups. Panel D is like a phylogenetic tree, a cladogram that highlights the evolutionary relationships between the significantly different taxa in the type 1 diabetes and the healthy controls. Basically, this image is a good snapshot of everything that's going on. And it shows that while the overall gut microbiome diversity is similar between the two groups, there are definitely some bacteria species that differ significantly, like the beneficial microbes like Faecalibacterium, which is more abundant in healthy individuals here. While pro-inflammatory taxa like, Prevotella capri are enriched in the type 1 diabetes patients. So in terms of the discussion, if we want to answer what did we know now and we didn't know before, we can say that, we know that adults with type 1 diabetes have a distinct gut microbiome, both in composition and function. And certain microbiome pathways are linked to blood sugar levels and lipid profiles. And we can attest that the microbiome alone can accurately classify type 1 diabetes versus healthy individuals, given that mean AUC number we had at 0.89. The broader implications, maybe the gut microbiome may serve as a biomarker for glycemic control in type 1 diabetes. It also highlights the need to integrate this data into clinical research, and, as the authors previously mentioned, ongoing studies will be needed to, make good conclusions in regards to this study ultimately. And this data hopefully will support the development of microbiome-based therapies, probiotics, diet, etc. Some of the unknown and the questions that we still do not know, how is this microbiome a cause or a consequence of poor glycemic control? If someone is suffering from type 1 diabetes, how does this microbiome lead to that? How does it regulate the glycemic control? And how do interventions targeting the microbiome improve type 1 diabetes outcomes? And other factors too, like genetics, immunity, environment, all those come into play. To conclude, this slide talks about or gives a visual summary of the key finding from Shallow et al. Study. I'm talking about adults with long-standing type 1 diabetes having a distinct gut microbiome, which is closely linked to their glycemic control. The diagram also illustrates a progression from individuals with type 1 diabetes through a unique microbial microbial composition, and ensures impacts of blood glucose regulation, and highlights the potential of the microbiome as a biomarker or therapeutic target for improving diabetes management. And ultimately, the microbiome is linked to glycemic control in adults with long-standing type 1 diabetes. Some of the questions that we can talk about and, think about as we review this article further, it's an ongoing study, and, it will be very interesting to learn about, more of the microbiome in relation to the blood glucose and how could diet or medications influence microbiome findings, genetics or environment. Um, it will also be interesting to do, um, you know, another experiment or research in a different country. Like I mentioned earlier, this was done in Israel, but I feel like it's a great model approach for a different country. And it will factor in the environmental difference between Israel and another country. And how does new technology like proteomics and genomics, how does that data, make the study insightful to study the microbiome host interactions in type 1 diabetes? There's a lot to answer, and I feel like it will be great to, indulge in a very fruitful discussion regarding these findings. And these are the references of the study.